Hi everyone, this is Arul Jothi from the Department of Biochemistry. In this video, we are going to see about the T4 bacteriophage virus. We'll see about the introduction, their characteristic structure, life cycle, and viral infection, and especially their role in cholera and diphtheria, and a few significances. Introduction. Actually, virus is an infective agent that typically consists of a nucleic acid molecule in a protein coat, and it will be able to multiply only within the living cells of a host. Viruses have few or no enzymes of their own, and they don't contain ribosomes. To multiply, viruses must take over the metabolic machinery of the host cell. That's why they are called obligatory intracellular parasites. Viruses have infected bacteria and they are known as bacteriophages. T4 bacteriophage is a virulent phage. It is also known as a lytic phage. T4 bacteriophage is a virulent phage. It uses the metabolic machinery of the host cell to produce the progeny viruses and kills the host in the process. Bacteriophage, it is a large and DNA virus. It infects the colon bacillus E. coli bacteria. It does not have a probacteriophage form. The T4 chromosome is approximately 168,800 base pairs long and contains about 150 characterized genes. Let's see the structure of T4 bacteriophage virus. As said earlier, it is a large virus with a complex protein coat. At least 36 different types of proteins make up the coat. Over 150 genes, 189 kb genome of DNA. The tail has an inner hollow tube called core. You can see in the picture. It is surrounded by a contractive sheath which consists of 24 annular rings. The distal end of the tube is connected to a hexagonal base plate with spikes or tail spin at each corner. Six long, flexible tail fibers also arises from the basal plate. The head is a capsid one. Then coming to the life cycle of the T4 bacteriophage in E. coli, you can see in the picture, the first step is the attachment phase. The phage attaches with the cell wall of the E. coli, then penetration. Following next to the attachment, the phage DNA is injected into the bacterial cell, leaving the phage coat outside. Then transcription. In this phase, they produce the mRNA, which is translated into the phage protein. Next is the replication of phage. DNA and synthesis of proteins will take place in this. Then fifth step is the assembly. The phage components are assembled into the mature variants. Then sixth stage is the release. The bacterial cell lyses and releases many infective phages. Then coming to the role of bacteriophage in cholera and diphtheria. Cholera is caused by the bacterium Vibrio cholerae. However, Vibrio cholerae does not cause the disease unless it is infected with a lysogenic T4 bacteriophage carrying the gene for cholera toxin. Cholera toxin is what which causes the severe diarrhea. Then Corynebacterium diphtheriae, the bacterium that causes diphtheria, it will carry a phage containing the gene for diphtheria toxin to cause the disease. Significance of T4 bacteria. These phages may be useful in killing bacteria that are resistant to antibiotics or in few cases where the antibiotic has difficulty in reaching the bacteria. These T4 bacteriophages are very specific. A specific phage will only kill a few strains of bacteria. Thus, they leave the native bacteria flora intact. T4 bacteriophage is also used in the recombinant DNA technology. In the past, these phages were also used by some to treat the infections. Thank you.